Definition for micropropagation is a way to make new plants using tissue or cell culture methods instead of natural seeds or buds. It creates plants with biotechnology without seeds, using methods like tissue or cell culture. In other words, micropropagation is the quick way of growing plants in a controlled environment with strong light-specific temperature control and a precise nutrient mixture. Our group have chose 4 example Malaysian crops in micropropagation. The first one is chili, or its scientific name is Capsicum annuum. Capsicum, a part of the Solanaceae family, progresses slower due to its strong reliance on genes and its stubborn nature. It's tough to work with in labs for cell growth, regeneration, and genetic changes, making it challenging to use DNA tech for enhancing its resistance to pests, disease, and tough conditions. Next is pineapple, Ananas communis. The pineapple plant is a herbaceous perennial and produces up to 200 flowers that join together to create multiple fruits, typically 15 to 20 months after planting. Pineapples, when fully ripe, can range in weight from 1 to 2 kg. Pineapple need biotechnology for development to fulfill market demand for fresh pineapples with exceptional sweetness, as well as uniformity and consistency in size and ripeness. Another example is papaya, Rica papaya. Papaya is almost entirely propagated from seed in commercial cultivation. The network's main goal was to create papaya varieties that could resist papaya ring spot virus disease and delay ripening process, which were the key challenges faced by the papaya industry. The last one is banana, Musa seminata. The banana biotechnology aimed at creating cooking bananas rich in provitamin A. Achieving this involved modifying banana fruit's carotenoid pathway by activating particular genes. The process has built a comprehensive collection of genetic data, encompassing genomic sequences and transcriptomes. Additionally, they have shown the capability to analyze these sequences, identifying potential genes for modification to instill disease resistance or improve agriculture traits.
Hey, you ready to the next contest? Let's go. Now, we continue with the basic concept of micropropagations. The technique of micropropagations provides a good alternative for those plant species that show resistance to practices of conventional propagations. An alternative method of vegetative propagations or mass propagations is offered through micropropagations. Plants in large numbers can be produced in a short period. Any particular variety may be produced in large quantities, and the time to develop new varieties is reduced by 50%. The micropropagation methods produce plants free of disease. Hence, disease-free varieties may be obtained through this technique by using meristem tip culture. For the in vivo propagations of specific plants, sexual reproduction via multiplications of vegetative parts is the only result, since they do not generate functional seeds, as seen in figs, grapes, bananas, and others. Successful applications of clonal propagations to the following is observed, like potato, apple, and many other ornamental plants.
Types of micropropagation. There are a total of 5 types of micropropagation. So the first one is meristem culture. Meristem culture is a method that involves in the cultivation of undifferentiated cells from the actively growing tips of plants in a sterile nutrient medium, leading to the production of genetically identical and disease-free plants. Next, collis culture. This is a method where undifferentiated cells or colors are cultivated from plant explants in a nutrient-rich, sterile medium, allowing controlled manipulation for kernel propagation and other applications. The third one is embryo culture. Embryo culture is a method of cultivating isolated embryos in a nutrient medium to foster their development into viable plants, offering advantage in breeding, hybrid production, and overcoming challenge associated with seed germination. Other than that, suspension culture. Suspension culture in micropropagation involves dispersing plants, cells, or tissue aggregates in a liquid nutrient medium, facilitating mass production in a controlled environment for applications such as extracting secondary metabolites or generating cell biomass. And the last one is protoplast culture. Protoplast culture is a micropropagation method where plant cells are isolated from their cell walls, forming protoplasts that are cultured in a nutrient-rich medium. This technique is essential for genetic manipulation and the regeneration of plants from modified protoplasts.
Now, let's go to the advantages of the application. The first advantage is rapid multiplication. This is focused on the crops that usually develop slowly or struggle to propagate by using traditional methods. When using micropropagation, it only uses a small bit of tissue to produce many similar crops quickly in an appropriate environment. The second advantage is pathogen-free plant. To maintain crop health and yields, sterilization of plant tissue and proper growth are key components of the process, which can lower disease risk and promote yield pathogen-free. Last advantage is genetic uniformity. In order to ensure the uniformity in characteristics like yields, quality, and disease resistance, micropropagation creates genetically identical plants that can lead to beneficial commercial production and consistency in crop performance. There are also disadvantages of the application. The first one is high cost. For some crops, tissue culture can be economically difficult effort due to high costs involving in setting up an operating facility, which requires specialized equipment, expert workers, and continuous demanding of resources operation. Next disadvantage is genetic stability. Genetic problems could give a result of somaclonal variation, a condition in which genetic mutation or changes happen during tissue culture, resulting in uniqueness among plants that are propagated. This could have an effect on the qualities of a plant that we wanted. Last disadvantage is cultivar suitability. Not every crop variety react to micropropagation in the same way. Some varieties might not adapt effectively in tissue culture environment, resulting in low success rate or low plantlet quality. In conclusion, Malaysia widely use the application of biotechnology like micropropagation to improve the quality of a variety crops and increase mass production. This application of biotechnology gives many benefits for the future of Malaysia's agriculture and the farmers by preventing losses in crops brought on by weeds, pests, and disease to improve harvest crop productivity and earn profit. 5 methods can be applied to conduct this process of micropropagation. The methods are meristem culture, callus culture, suspension culture, embryo culture, and photoplast culture. Examples of plants that use micropropagation are papaya, banana, chili, and many more. That's all from us, and these are the list of reference that we use to research in our presentation. That's all from us. Thank you.